let's move on to demonstration. Okay, so I will take aspirin, okay, aspirin and its derivative, and I will prepare a model based on the, you can say, biological activity of aspirin, that is COX receptor, as I've already told you. So it has an analgesic property, and it fits into the cyclooxygenase receptor as well. So let's do that. Okay, let me share another screen. So just a minute. Okay, and let me share. Okay, so here is the, okay, let's see the demo first. Okay, so in here I have selected 16 different compounds. Okay, aspirin and indomethacin are FDA approved drug, and these all are the compounds I have obtained from Campbell database. Okay, and they do not have their name because they are in clinical trial. Okay, so in different stages of clinical trial, 1, 2 or 3. Okay, so I have obtained those compounds. So actually the my test compound has a scaffold of aspirin. Okay, so if I am testing any kind of compound molecule that is related to aspirin, then I need to prepare a model based on the scaffold. Okay, so aspirin is a scaffold here. Okay, so aspirin and their derivatives, I can easily you can say collect those compounds and then prepare a model. Okay, so that's the basic idea. So in case of aspirin, the PIC50 value. So PIC50 value is a different thing than IC50 value. Okay, we can calculate PIC50 value using this formula. Okay, so later on I will let you know about it in the Campbell as well. So in the Campbell you can see the standard, you can say standard value of any kind of compound molecule, and based on that standard value you just need to calculate the PIC50. Okay, so later in another column you can see the P Campbell IC50 value, that is supposed to be PIC50. Okay, you can either copy that PIC50 value, or you can even convert through the standard given over there. Okay, so you should not write the units here. I've just used this unit here, but you shouldn't write it because PIC50 doesn't have any kind of units. Okay, so I've just copied the bioactivity of these compounds as well. So the bioactivity needs to be similar. So these all have the bioactivity, similar bioactivity, same bioactivity, actually not similar, same bioactivity. And next is target, so they all target on the same receptor, that is cyclooxygenase. Okay, so the PIC50 value has been obtained over here. So let's move on to the molecular descriptor now. So I need to allocate some of the molecular descriptor of these compounds. Okay, so aspirin, indomethacin and so on. So here you can see, 16 different compounds and a small string as well, and the molecular descriptor of those compounds has been given over there. For example, I have taken molecular weight, log p value and topological surface area. I've told you about selection of you can say molecular descriptor. So I have selected on 3 different categories. Okay, for example, log p value, it signifies is the lipophilicity of these compounds, that is directly linked with fat solubility, and that determines the membrane permeability, and it also plays role in absorption, distribution, and even not metabolism, but particularly absorption and distribution. Okay, and in case of molecular weight, so molecular weight directly indicates the size of any kind of compound molecule. So it actually acts as a you can say indicator of permeability. Okay, permeability, or you can say penetration. Yeah, that's a permeability. Okay, so the larger molecules are less permeable and the smaller molecules are easily permeable to the cell membrane. And next is topological surface area. So topological surface area, it it signifies as the area covered by polar atoms of that particular compound, and it particularly indicates absorption, blood-brain barrier indicator as well. It is an indicator of blood-brain barrier as well. And next is it also signifies the hydrogen bonding activity of that particular compound. Okay, hydrogen bonding activity of particular compound. So these has different roles. Okay, so based on these roles, I have selected this parameter. Okay, you can select number of different molecular descriptor, but it has certain you can say guideline as well. For example, the number of compounds. Okay, the number of compounds that you are going to prepare a model is greater than or equal to 5 into molecular descriptor, number of molecular descriptor. Okay, for example, here I have used 16, or you can say 15, 16 compounds. Okay, 16 compounds, and there are 3 different molecular descriptors. So 16 is the compound number of compounds. 16 is greater than or equal to 5 into 3, right? 5 into 3, that means 15. Okay, obviously 16 is greater than or equal to 15, so it satisfies that particular you can say guideline as well. Okay, so based on that you can select the number of compounds. Okay, how many compounds do I need to select would be done through the number of molecular descriptor. For example, I've already told you about it. Let me make it clear as well. 5, or you can say number of compounds is greater than or equal to 5 into number of molecular descriptor. Okay, so that's where you can select you can allocate the number of compounds as well as a number of descriptor in your particular model. Okay, and next is I've copied the PIC50 value from the Campbell. Okay, that was already shown as well, and I've used the source as well. And later I plot that, or you can say I calculate the regression analysis, or I have done the regression analysis of these compounds using PIC50 value and molecular descriptor, and I've got the result. So in x-axis you can just plot these molecular descriptor, and y-axis, or you can say as a y component, you can use PIC50. Okay, so you can go to data. Okay, if you do not have data analysis, in some cases if you have not used data analysis before, then you will not get that. So you can just go to file, and you can go to options, and you can go to add-ins. Okay, and then you can go to Excel add-in, go. Okay, you can get a here you can get the analysis toolpath. You just need to select it. Okay, you just need to press OK here, and you will get the data analysis toolpath there. Okay, so in this way you can do that. And next is to get the information of regression, you just need to go to data analysis. Okay, and then you can go to regression. Okay, you can go to regression, and then you can just in y-axis you can just select this. Okay, till here, and then in y or in x-axis, or in x-axis you can just select molecular weight to topological surface these data. Okay, in y-axis you need to select PIC50, and in x-axis you need to select this molecular descriptor. Okay, once you do that, you will get that result. You just need to allocate the output range as well, wherever you want to put the summary output file. Okay, you just need to allocate. For example, if I have allocate here, so I've got the result. You can do it in a similar way. Okay, if you press OK here, then you will get the similar kind of similar kind of output file. Okay, so here you can see. Okay, so let's talk about this data right now. So here the regression statistics or the output file has been generated, and the R square value is quite good. Okay, 0.83, and the adjusted R square. So we need to look for the adjusted R square actually, not R square. Okay, so adjusted R square means it has already, you can say it has already analyzed the data based on number of descriptor and number of, you can say, lipophilicity, or number of, sorry, PIC50 value. Okay, so if you have enough data over there, you will get a nearby you can say R square value. Okay, it is an adjusted R actually, so the value needs to be similar. Okay, for example, here I can see 0.83. Okay, and next is 0.79. It is close enough. So if you have not selected the, you can say, proper molecular descriptor, number of molecular descriptor, as I've already told you, the number of compounds should be greater than or equal to 5 into number of a number of molecular descriptors. So if you haven't satisfied that thing, then you will not get a R square, adjusted R square in this kind of format, or in this kind of ratios. Okay, so the ratio needs to be exactly feasible to the equation. Okay, so that is the thing. And next is the significance. Okay, significance needs to be 0.005, less than 0.005. It is quite lower, so the statistical it is significant because we have taken enough data. Okay, so that is the thing. And next is intercept. You can get the value of intercept over here. So I have copied the intercept value here, and then the coefficient of variable x variable 1. So in case of my data, x variable 1 is molecular weight, x variable 2 is log p value, and x variable 3 is topological surface area. So I've allocated them based on the information given here. So x1, x2, and x3. Okay, so we need to use this information to calculate our you can say calculate our predicted IC50 value. Okay, so let's go to calculation now. So here it is. For example, in my data, I have used 16 different compounds, and the actual IC50 values has been given here, and the predicted one. So predicted one, we need to use this equation to see how the data has been, or you can say distributed. Okay, so the IC50 value of PIC50 value of aspirin is 4.82. Okay, so once I prepare a model, I found out that the IC50 value of aspirin is 5.02. Okay, so how did I calculate it? So let's talk about it. So the formula is here. Okay, PIC50 value. To predict PIC50 value of any kind of compound, we use this formula: A plus B1 x1 plus B2 x2, and so on. Okay, so there are three different you can say molecular descriptors. So x1, x2, and x3 are satisfied over here. A stand for intercept, so 3.1, and B1 stand for the coefficient of x1, you can say molecular descriptor, and B2 stands for coefficient of x2, and B3 stands for coefficient of x3. Okay, so in this way we calculated it, and the aspirin has the predicted value of 5.02, and so on. Okay, you can see the values lying over here. Okay, and we just need to plot this plot the scatter diagram as well, scatter plot as well. So to plot that, you just need to place actual PIC50 value on x-axis and predicted value on y-axis. Okay, and you will get this kind of information. Okay, so if the regression is fine, okay, it's r square is 0.83, and you can see okay, you can see the ideal model. Okay, in case of ideal model, the x or you can say x value needs to be equal to y value. Okay, that is the ideal one, but actually we do not get ideal one, but the value, or you can say the chord needs to be diagonal in shape. Okay, so it's quite you can say matching as well. So I can easily say that my model is better. Okay, so in this way we can allocate, we can predict or you can validate our model as well. Okay, next is you need to plot predicted as well as residual IC50 value as well, PIC50. So to calculate residual IC50 value, you just need to okay, you just need to calculate the difference between actual and predicted one. Okay, so actual minus predicted, you can get the value over here. Okay, it is not nanomolar, sorry, we cannot use the unit in PIC50. So in this way you can plot it. Okay, and the ideal curve says that the value lies in the values should be in a in this zone. Okay, 0 zone, or you can say the value lies in this linear axis of 0. Okay, so it is quite fine as well. Okay, ranging from minus 0.6 to 0.6. So next is, okay, you can also correlate your data. Okay, so correlate your IC50 value with molecular descriptor actually. So I have done one correlation analysis. Okay, I've checked whether the log p value directly impacts the actual IC50 value or not. Okay, so in this case, okay, I plot log p value on x-axis and actual IC50 value on y-axis, PIC50 value on y-axis. Okay, so after doing that, I got this information. Okay, so actually this information signifies that log p alone cannot say that, okay, the value of log p cannot determine whether the compound is you can say active in that particular receptor or not. Okay, for, because you can see the regression is very low. Okay, so we cannot say that log p has that important role for change in IC50 value of any kind of this particular compounds. Okay, so you can even plot for you can say what we have done. Okay, molecular weight. Okay, next is topological surface area and so on. Okay, any kind of molecular descriptor that you have used. Okay, you can just plot that. Okay, and then see whether it is correlated with your predicted IC50 value or not. Okay, in this way you can just link these kind of thing. Okay, and if you have a confusion like, okay, so IC50 log p has no any impact on increasing or decreasing value of PIC50, then you just need to think that, okay, log p is a sole parameter. It doesn't it didn't signifies the role of this compound in cyclooxygenase. But if you consider the number of different molecular descriptors, then you can see it has been impacted. Okay, and we can also get the data from here as well. In actual IC50 it's 4.82 and predicted it's 5.02. That means it's quite similar. Okay, so in this way you can validate the data as well. And now talking about how you can use this model to analyze your compound. Okay, for example, in my analysis I have taken this compound. Okay, it is a aspirin derivative. Okay, so I want to know whether this derivative is active against cyclooxygenase or not. Okay, so to find out the thing, I use its molecular descriptor, 3 different molecular descriptor. We need to use exactly the same molecular descriptor that we have prepared in our model. Okay, so I have used molecular weight, log p, and topological surface area. Okay, so these are the data of this particular derivative. Okay, I can get this from databases like PopChem or Camel even. Okay, so I have used this descriptor here, and I calculated the PIC, predicted IC50 value, PIC50 value of, or you can say predicted IC50 value using the same regression curve. Okay, the A value is similar, B1, B2, and B3 are similar. That has been obtained from, you can say, the model itself. Okay, so after calculating it, I found out the PIC50 value was 7.94. Okay, the PIC50 value was 7.94. So to see whether my compound is active against cyclooxygenase, I need to go through the same data. Okay, to check the value and same data. So it was 4.9, 7.9, sorry, 7.9. So you can see the actual IC50 value first. Okay, 4.8, 8, 6.1. So my compound falls in between, falls in the range of indomethacin. Okay, that means it has a great binding activity against cyclooxygenase. Okay, because the PIC50, the greater the value of PIC50, the more binding nature, or you can say more biologically active the compound is. Okay, so in this way you can check whether it is feasible or not. So in case of IC50 value, the lower the value, higher is the activity. Okay, in case of IC50, but in case of PIC50, the higher the value, higher is the biological activity, because it is calculated in the negative logarithm form. I have already told you. So if you have log p value, if you have IC50 value in molar, then you can easily use this equation to calculate PIC50. Okay, so this is how we can predict the biological activity of any kind of derivative. Okay, so let me open this Campbell, so it will help you out. Okay, it's a Campbell database interface, so I have used this already, so you can get the results over here. So I have searched cyclooxygenase in this search engine, or you can say space. Okay, and then I got this information. So in the left hand side you can use different filters. Okay, you can use the filters, and then you can easily get the information. Like on the left, on the right hand side, you can see a number of different compounds. Okay, and then the IC50 value has been given over here. You can just go through it. Okay, like uh, let me say select. Okay, let me see. Okay, you can see the IC50 value over here. Okay, where is the standard? Okay, standard value is present over here. IC50 value 89. So it's IC50 value. Okay, it's not PIC50 value. But here you can see a P Campbell value. This is PIC50. Okay, 7.05 is a PIC50 value. So in this way you can just copy this. Okay, and about the assay, you will get the information of assay here. Okay, assay description. You can see inhibition of human kidney, whatever thing. Okay, selectivity. Okay, you can easily get this two IC50 value from this particular interface. Okay, you cannot just copy the IC50 value of this one and this one. Okay, because the assay are different. Okay, so in this way you can select it. For example, assay format, here it is assay format, it is a protein format. Okay, you can go onto the left hand side and you can you can check it. Okay, assay format, you can just put a filter over here. There are three compounds for assay format. Okay, assay format, you can, but the thing is these two are similar, but the assay description here has been given different than the rest two one. Okay, so you cannot select this along with this two. Okay, so that is the another clue for selecting how you can select compound and IC50 value by using Campbell. Okay, so in this way you can do it. Okay, let's prepare one model. Okay, you can use for practice you can just use 3 or 4 compounds. Okay, forget about the significance of data. You can just copy 3 or 4 molecular descriptor or three or 4 compound, and then you can check it. Okay, so let's do it. So I'll give you some time. You can just go to Campbell and search the biological target of any kind of compound molecule that you are interested in, and you can just copy it. Okay, so let's do it.